Okay, so the next company is Alzecure. We have CEO Martin Jönsson. Welcome, and please take it away.
Thank you so much. Hello, my name is Martin Jönsson, and I am the CEO of Alzecure Pharma. And I'll give you an update on who we are, our focus areas, our projects, plans, and also progress. And the last month for us has been very eventful, so I'm very happy to be here and report on this. We have presented new positive phase 2 data in our pain trial, ACD440. We have published 3 new publications, and we have also seen progress in all our projects and also in our categories. And we will also, in the coming month, present additional data. Just a brief background on Alzecure. We focus on Alzheimer's disease and pain. We are a spin-out out of AstraZeneca, started our company when they left the CNS field. And we have been sponsored by the Swedish Alzheimer's Fund, who are still a shareholder in us. We are based at Karolinska Institute, and we work very much with Karolinska and more. And we only work with small molecules. And we have 3 different platforms. Alzstatin, which is a disease-modifying preventive treatment against Alzheimer's. And then for patients who have developed cognitive disorders and Alzheimer's, we have Neurorestore to help individuals to improve learning and memory capabilities. And here we now also see data on potential disease-modifying abilities. And then we have our pain platform, where we have 2 different projects. And we have been listed on NASDAQ since 2018 and have a market cap of around $250 million. And this is our pipeline that we are very proud of. In Neurorestore, we have ACD856, track PAM, where we are focusing on Alzheimer's disease, but also running projects around depression. And we last year published positive data in a phase 1, showing safety, tolerability, and also target engagement. And now, during the last month, we have been publishing new data on disease-modifying abilities. And on Alzstatin, our disease-modifying preventive treatment that we are developing, we this year came with a new molecule, an additional molecule, and with this we hope to have an even better molecule, but also longer patent life, which is very important. And then in painless, we have ACD440, our TRPV1 antagonist, where we have presented positive phase 1 data. We did that in May, and now 2-3 weeks ago, we presented more detailed data from this trial. And on all our projects, we are working with academia and experts in the field, KOLs, like Bengt Wimblad, Maria Eriksdotter, and Henrik Zetterberg. And if we look at our Alzheimer's platform, where we have our main focus, we have Alzstatin, and here we are focusing on diminishing the production of the toxic protein which builds up into plaque, Aβ42. And then Neurorestore, our track PAM, improving memory and learning capabilities. We have Neurorestore to help this pool of individuals. And what's very good with both our projects is that they are validated by global leading companies like Roche, who have a project as a gamma secretase modulator, and then Eisai, who has a track A PAM. And we will get back to this because much is happening in this field, and this is truly helping us. Alzstatin, here, as you know, over time in patients developing Alzheimer's, they have built up plaque, which is destroying the neurons. What we are focusing on is the main building block, which is starting this cascade, Aβ42, and we are able to diminish this production with over 60%. And when it comes to this approach, gamma secretase modulator, it is an approach which is getting more and more noticed. If you were at ADPD this spring, they talked about it. Now they are also publishing more and more on it. What's great here is that true advances are now being made. Roche, who have a project in this, it's more or less only we and Roche who are present here for the moment. What they are going to do now on CTAD is that they will present clinical data on a trial in phase 1, and they will also present data with input for their phase 2. So things are really moving in this space, and that we also are feeling with the discussion of potential external parties. So with this data, it validates what we are doing, it's de-risking what we are doing, and it's also giving on guidance with regard to how to approach our project. So this is very positive for us. And what's also interesting with this project is that we already in phase 1 will be able to show proof of mechanism. Because in phase 1, we will be able to show how we can diminish the production of A-beta 42, how we can lower it and the effects. And based on this, we will hope to see that we have a very good molecules and also very strong value driver with regard to out-licensing. And to identify the patients which are relevant, we use blood test and also biomarker kits. And all of this already exists. And when it comes to the potential patient population here, there are multiple patient populations. We see the opportunity to use it as a maintenance treatment in patients who have developed Alzheimer's disease to keep down their amyloid load. And then in combination with antibodies. And then also over time to use it as a preventive treatment to making sure that people do not develop Alzheimer's disease or that it's delayed. And then to Neurorestore, to help individuals learn and remember. Here we have already shown in preclinical model that we have the ability to increase the ability to learn, to store what you learned, but also to remember what you have learned. The way we are doing this is that we are using our molecules, Neurorestore molecules, which are track PAMs, increasing the signaling of BDNF and NGF. And what we now have shown as well in multiple different models are that it looks like that we also have a disease-modifying ability, that Neurorestore HD856 is neuroprotective against A-beta. We also induce neural outgrowth. We improve synaptic health, and we also increase mitochondrial activity and function. So we see this not only as an opportunity to improve learning and memory capabilities, but also potential disease modifying. And we are not alone when it comes to believe this. We are privileged to have ASI also driving a project in this, but they have a track PAM, which is a bit different, only driving NGF signaling. And they have gone out and communicated, this is a quote, that they see that their project, A2511, has a can be a disease-modifying therapy in neurodegenerative diseases. So this is general. So Alzheimer's disease, Parkinson, frontal lobe dementia, etc. And what they are doing is validating also what we are doing and also what we are showing. And they will now present new data, new clinical data at CTAD. So this is very good for us. And we will also present there. We have a poster there on showing additional data on neuroprotective and neurorestorative effects. So this is very, very exciting for us. On track A-NAM, we have also during the summer published data in depression, but we don't have time to go into that in detail, but that is also a field where we are seeing more and more interest. Then to our pain platform, where we have track A-NAM and also ACD440. And ACD440 is in phase 2. And here we have a TRPV1 antagonist. Neuropathic pain is the individually biggest pain area. And our project is building on what in 2021 was awarded the Nobel Prize in Medicine. So ACD440, a TRPV1 antagonist, initially developed by AstraZeneca, and then we have the privilege to take it over. We have developed it into a topical formulation, a gel. We then did a positive 1B study and showed safety tolerability. And now we also came with positive data in peripheral neuropathic pain, 2A study. What we have shown is that we show proof of mechanism in patients with peripheral neuropathic pain, and this in patients who have chronic pain. And we are showing that we can decrease pain with around 50%, which is seen as clinically significant and relevant. And we also show that the substance and the formulation is well tolerated and safe. And all of this data supports both continued clinical development, and here we are focusing on out-licensing, but also driving potential projects going forward. And then key investment highlights. Why is Alzecure an interesting company to invest in? Because we are targeting areas of significant medical need. Also, we have a strong team with extensive experience and track record. We have been able to identify targets, develop molecules, run them through preclinical and into clinical and up to phase 2. We also have platforms with first-in-class properties, which really can become game changers. And also that we are doing parallel investments with follow-up programs. And also that each of these projects can be blockbusters, multi-billion market opportunities. And also that we now are a company which is in phase 2. So with that, thank you so much for your attention, and please go to our website to learn more.
Okay,
Yes.
thank you very much, Martin.
Thank you, Frank.
Just interesting because we had Oblique here on stage an hour ago, and they have an antibody,
Yes.
and you have a topical gel.
Yes.
It would just be interesting to hear how they differ and what the advantages with your strategy could be.
Yeah, first of all, about 60% of these patients have upregulated, how should I say, receptors in the skin. And to be able to reach them through a gel is a great advantage. What we have seen with regard to other formulation going oral is that they are hitting systems where you have side effects. And also what's very good with our molecule is also that it can be used in not only gel, it can also be used in our other formulation. As you maybe know, Novartis has presented or done a phase 2 in ophthalmology, in neuropathic pain in eye. So we also see the opportunity to develop ACD440 for that kind of formulation. But also based on the data we have, we see a significant, how should I say, effect, but also very good safety. So this is very, very good.
And you mentioned the phase 2A results. Could you maybe just elaborate a bit on those, and what type of treatment effect or effect size did you show, and what does it mean?
Yeah, so the patients that we had in the trial were chronic patients. They have had neuropathic pain for a very long time. We even had patients in the trial who had been on opioid and hadn't been helped. And we actually had some patients who were on low-dose opioids. We also had patients who were treated with several other oral medications, but still they had a problem. When they used our formulation, we could see that we could reduce the experienced pain with up to 50%. And generally, when it comes to chronic treatment, they say that if you can go above a treatment effect of 30%, then that's clinically relevant. So what we see is a very good effect. We show proof of mechanism and target engagement. And also, as I stated before, very good safety and tolerability. So no patient whatsoever dropped out.
And in a follow-up study, how would a trial design differ? I suppose that this was induced pain, right? And I would assume that you would look more at spontaneous pain, or how would it differ?
Yeah. So we didn't. These patients, of course, had chronic pain, but the study that we will do, a so-called proof of concept, would be, how should I say, a much bigger patient population. And based on this, to select the patients, we now have a bedside kit, so you can really test the patients as well to see which patients will probably respond. So we had a pinprick, we had a brush, and also testing the patients with regard to cold and heat. So this will help us to identify the patients who will respond. And this is a de-risking of the clinical trial. And then what we are looking at now is, of course, the size of the trial. And we have several different, how should I say, possibilities there. And then we are looking at some other potential trials and indication areas as well, because we don't. You both have acute pain and you have chronic pain, and we are looking at how to approach this.
Okay, so the selection, you could use the pinprick to select patients.
Absolutely.
Yeah, but it would it be to show an effect in this other patient population in the phase 2b trial? It still would be a larger patient population, I assume.
Absolutely. And what's interesting here is that we showed effect already after 1 week. And we believe, if we look at pain trials normally, that you need 4 to 6 weeks before you can see, often to see a significant effect. So we are looking forward to a longer trial where we really can show the true potential. And potentially what we also will see is that there will be some patient population additionally which will respond to ACD440.
Interesting. And if we move over to Alzecure,
yes.
You mentioned early proof of concept.
Yes.
What does it mean? What type of results would it be? Can you explain that?
The patient that we would have, we will be able through blood samples to see how we can reduce the production of A-beta 42 and 40. And this is the effect that you also are looking for in patients, so to say. So we will be able to see that kind of effect.
And in a pivotal trial, what type of endpoints could you be able to use then? Would it be to look at amyloid beta production, or would it be some sort of measure of patient outcomes like today's Alzheimer's trials, or do you know?
That depends a bit on. First of all, you need to have an efficacy measure that the payers are willing to pay for. So that we need to look at. But we also know that the regulators are also stating that based on biomarkers like A-beta 42, you could potentially also have an approval. And here we think we will look at amyloid load, to what extent we can affect the amyloid load in the patients. So initially we would go into patients who have developed Alzheimer's disease, and maybe to see how we could keep down the amyloid load in patients who have already received antibodies. Then the benefits of combining an antibody and alstatin, this is a concept that Eli Lilly has been publishing on with regard to how to use this approach. And then the end goal is really to treat individuals who have not developed Alzheimer's disease, but who we see are at risk, and based on their genetics being ApoE4, and also that we see that they have a high load of A-beta 42, the protein which is building up into plaque and destroying the brain.
And if we move over to ACD856, and you mentioned a competitor or a peer from Eisai. Could you explain a bit more on how they differ from each other?
Yes. So Eisai have a track PAM, and to work on the track A receptor means that they are affecting the NGF signaling. And NGF is important for brain health in general. Then you have the neurotrophin BDNF, which is the track B. And Eisai is only working on track A, NGF. We are working on both track A and track B. So increasing the signaling of BDNF. And BDNF is very important when it comes to cognition. But BDNF is also important to brain health with regard to neural outgrowth and so. And we will publish data showing what the benefits are to have both of these activities. And we are running trials where we are looking at only having track A activity or also having track B activity. So we have data on these different avenues and possibilities. And we are a strong believer in that you should have both. So both track A and track B induce activity.
Very interesting.
Yes.
I think that's all the time we had. So thank you very much, Martin, for being here today.
Thank you, Frank. Thank you. Thank you.